Okay. Mrs. Erickson right here. We're going to go through non-coelomate invertebrates. Now I know it says chapter 33, but our new edition, and I'm going to have to put these notes out to pasture, but this new edition actually breaks it down into either deuterostomes or protostomes, which I think is a lot easier for organization-wise. So what I did is I'm going to present it in this view because in the notes over here I actually have page numbers in the book that you can find the organisms and actually read a little bit more about them if you're struggling to understand. But non-coelomate invertebrates. Okay, so traditionally this is what the animal tree kind of looks like based on morphological information. Where you have the parazoas, the sponges here, and our true tissues over here, and then you break it down into the body plan, radial versus bilateral, and then we get into the acoelomates, which means no body cavity, 2 body cavities, and then from there pseudocoelomates, which means fake body cavity, and then our coelomates, and then even further break it down into protostomes or deuterostomes, and that's the way it has been. But after, with molecular data and all that, now we've kind of changed it to this, parazoas versus eumetozoans, and we go radial bilateral, protostomes versus deuterostomes, this is kind of like the format your book has broken it now down into, which is nice. And like I said, it's my last year with these notes, so we're gonna be kind of jumping around a little bit. Actually, we're going to cover the Prophora, Cnidaria, Ctenidophoria, Platohelminthes, Rhodophoria, Nematodes. I can't read that one, and we'll probably stop like right here. So we're going to cover a lot of organisms today. Okay, so the revolution in invertebrate phylogeny. Remember, invertebrate means no backbone. Okay, so traditionally, we used symmetry in the body cavities that we saw. With their new phylogeny, protostomes are made of 2 clades, the spiralians and the ecdysozoans. So I'm going to talk about these guys first and then kind of jump into body cavities. So with the spiralians, it just means that they have basically spiral cleavage. Most of the spiralians live in the water. They propel using cilia as well as some body muscular contraction to move or to bring food in. So there's going to be 2 subgroups, and the first subgroup is called the Lophotrochozoa, a coelomate, true body cavity protostome. So mouth forms first, has muscular contractions, has a special type of free-living larva called a trochophore. It does include other phylums that we'll talk more in depth tomorrow, I believe. The bryozoas, the brachiopodas, the mollusks, and the annelids. So just know Lophotrochozoas are spiralians. The second subgroup is the Plato Zones. Okay, mostly acoelomates. They do not have a body cavity surrounded by a mesoderm. These guys tend to be flat. They move by ciliary action. Some have some complicated jaws, as you can see here with the parasite, the tapeworm. It does include the phylum Platyhelminthes, which I will discuss more in depth later on in this lecture. Very simple body plan you'll see, but it also includes the flukes and the tapeworms. Now, the ecdysozoans, okay, a little bit more complex. How they grow in size is through molting of their external skeleton. And so when we get to, what is that lecture called? The coelomates, part 1, I believe we'll talk about the molting of mollusks and arthropods, okay? There's a lot of phyla underneath the protostomes, the nematodes and the arthropods. Again, I think today I will talk about the nematodes, okay? Because of this, they have a pseudocoelomate, which is a fake body cavity, and I'll explain what that is when we get there. But then the arthropods are part of coelomates, and that will be part of our coelomate lecture. So this is based on molecular comparisons, okay? This is based on body plans. And so they color-coded it to kind of show you how things get shuffled around a little bit. So our new book breaks it down pretty much this way. The old book, which is what these notes are based off of, will be covering this half right here. the radia, acoelomates, and the pseudocoelomates, which are also partly bilateral. So just another family tree showing the big basic phylums of animals. And we're going to be, I just talked about the ecdysozoans and the lophotrochozoans, and so I'm just trying to show you this is where they are. They are both protostomes, where the mouth forms first. But anyways, enough about ecdysozoans and lophotrochozoans. Let's kind of jump into sponges. Where are they? Down here. So we're going to start here and kind of work our way this way, and then we're going to hop over a big group here and then continue on over here. Okay, so with parazoans, these are animals that lack specialized tissues, okay? But they are considered part of the animal group because some cells are specialized. They have cells that are specialized to rapidly contract, they have synchronized reproduction, and they do build some kind of complex organized meshwork of structures called spicules and spongin. But few are radial symmetry. In fact, a lot of sponges don't even have symmetry, okay? And another thing that they have is that they can move, but it's their larval stage that are the free swimming, and when the adults become anchored to a structure, they don't move after that. So the body is composed of several cell types, okay? We have a wall made of basically 3 layers here. The Cnidarians face internal, so inside here, this is the Cnidarians. We have the outer epithelial layer of the flattened cells, so it would be the kind of like this dark orange color, and then the mesohyl, the fill-in in between. Sponges can reproduce asexually as well as sexually. Now, what do spicules and spongins do? Spicules are kind of the silica, the needles, the gives it texture, if you will, and the spongin is just tough protein that kind of interfills the spicules. So you can see the spicules are embedded inside the spongin, but both of their function is to strengthen the body of that organism. Now, zooming in on the Cnidarians here, very similar to the protist Cnidoflagellates, you can kind of see there's this flagella here, and it moves and it filter feeds. So it creates kind of a current where water is moved in to the organism, and they filter feed, and then water gets pushed out through the osculum, which is the opening on the top side of the sponge. So that's all I want to say about the parazoans. Moving on to eumetazoans, animals with true tissue. So if with animals with true tissue, we tend to have some complex body plans, an ectoderm and an endoderm. So remember, your outer ectoderm usually is like the epidermis layer, the skin layer, as well as your nervous system. And then the endoderm is usually your digestive tissue. Okay? If you are bilateral, if you are a bilateral organism, you do have a third germ layer, which is the mesoderm, that forms the muscles, okay, and the bones. So some organisms just have 2, we call them diploblasts. Some organisms have 3, we call them triploblasts. But there are 2 radial symmetrical groups that are around today, the Cnidaria and the Ctenophora. Cnidarians are your jellyfish, your sea anemones, your corals. The Ctenophora are your comb jellies. So I'm going to break it down, starting with the Cnidarian, their basic body plan. 2 basic body plans, polyps and medusae. So the polyp means that it kind of becomes anchored, attaches to some type of substrate. They are cylindrical. The body opening acts as both mouth and anus, and it faces upward. The medusae is almost like a tiny little, it looks like a jellyfish, okay? It's free-living, umbrella-shaped, does have tentacles surrounding the mouth. So here's the medusae, here's the polyp, and here's the medusae. Now the life cycle can occur as polyps or medusae, or they can actually alternate between the 2 phases. But I just want to emphasize that both polyps and medusae are diploid, okay, 2 germ layers, diploid. Polyps can reproduce sexually or asexually, and medusae is just sexually. Now the basic body plan of Cnidarians is that they do have internal extracellular digestion of food. So the mouth acts as both the mouth as well, I guess. So this opening acts as both the mouth and the anus. So it will bring that organism in, and undergo some internal extracellular digestion and then excrete the waste products out. And this is a huge major evolutionary innovation because now they can eat animals that are larger than the individual cells. Whereas with sponges, they filter feed, so it's smaller than the cell. Now, with Cnidarians, they do have some specialized cells called Nato Sites. Okay, and the Nato Sites basically are found in Nemocysts. It's almost like a harpoon. So this harpoon's coated with toxins. The barb will hold the prey, the toxins subdue the organism, and then it's brought into the mouth and ingested. So here's the tentacle of our Cnidarian, and if we zoom in on that tentacle, you can kind of see that we have a nemocyst, or nematocyst here, and inside that is the cnidocyte. So there's this harpoon, it gets ejected, the toxin gets into the prey. So yeah, this video I think is just very, well, it's not going to play for me. All right, well, moving on. There are 4 classes of the Cnidarians, and you know what? I forgot to emphasize this. You can find Cnidarians on page 655 through page 651 through page 655 in your book. Okay, so there are 4 classes of the Cnidarians: Anthozoas, Hydrozoas, and Scyphozoas, as well as the Cubozoas. And so if you'd like to read some more information here, there's a link, but again, your book actually does a great job going through each of the 4 classes of Cnidarians. Okay, so that's the Cnidarians. Moving on to the Ctenidophoras, comb jellies. Okay, comb jellies. Okay, so basically the Ctenidophoras, you can find this on page 655 to 656. Not a lot of them, but they don't have nematocytes. They don't have those stinging cells that can, subdue their prey and bring it in. Okay, but their structure is a little bit more complex. They do have an anal pore where water and substances pass completely through the organism. The Ctenidophoras tend to be transparent, and they only have 2 retractable tentacles. What they do is they capture prey with a specialized structure called the coloblast. So the prey makes contact, the coloblast bursts, and discharges a very strong adhesive material. So these are what the comb jellies kind of look like. You can kind of see that they only have 2 tentacles here. Water propels through the organism through 8 rows of comb-like plates fused with the cilia. These guys tend to be bioluminescents. They are diploblasts with radiosymmetry. So that's really all there is about the comb jellies. Moving on to bilateral symmetry and acoelomates. These guys do not have a body cavity. There's no body cavity in the mesoderm. Okay, and then I'm going to talk about the pseudocoelomates, which will be orange background, and pseudo means fake. If you look over here, they have a fake coelom. The cavity is partially lined by the mesoderm. And then chapter 34, or actually I'm going to break this lecture up into 2 parts: coelomates part 1 and coelomates part 2. So this diagram shows the acoelomates, and you can see that the body cavity is not covered by the mesoderm. And here it's partially covered, and then here it's completely covered. So first up for acoelomates, no body cavity, is the platyhelminths, which are flatworms. Very soft body organisms, flattened dorsal ventrally. They do have a definite head, so we do have some cephalization going on here, and that makes sense because they're bilaterals. A lot of them are parasitic, but they can be carnivores and scavengers, and move by ciliated epithelial cells. Now, digestion, they do have a digestive cavity with an incomplete gut, basically only 1 opening, so they cannot feed, digest, and eliminate simultaneously or continuously. So they'll eat, they'll digest, and then spit it out, and then repeat. The mouth is located ventrally toward the mid part. They do have muscular contractions, usually associated with strong sucking force, so it can ingest food and tear it to bits. Their gut is branched, and it extends throughout the body because when you break down nutrients, then cells need that, and so it functions as both digestion as well as transport of food particles. And so we have some phagocytosis going on here. Tapeworms do not have digestive systems. They just absorb the food directly through the body walls. And if you just kind of think about tapeworms, where you find them, that would make sense. Now, excretion and osmoregulation is a little bit different, unlike the Cnidarians. They do have these tube networks that run throughout the entire body, and we basically call them, I think, flame cells. I'm looking for the word here. thought they were called flame cells. Maybe I'm ahead of my, oh, there it is. Tubules have flame cells that move water and excrete substances into the tubules, and then they exit through the pores. Okay. For organ systems, no circulatory. Okay. They do have a nervous system made of cerebral ganglion, just a bunch of nerve cords that run down the body. And they do have eye spots that are very sensitive to light, so they can distinguish between light and dark, and they tend to move away from extremely strong light. Now, how they reproduce, a lot of them are hermaphroditic, which means that they both have male and female sexual structures. But here's a little comic strip for you. So tuberella flatworms are hermaphroditic, with viable male and female sex organs. Pretty great, I know. But because of this, when it comes to reproduction, who's going to be the mother is up for debate. And it turns out that being pregnant is hard work. So these worms fight it out. And by fight it out, I mean they pull out their dagger-like penises and each try to stab sperm through the other's flesh. The loser gets pregnant first. The loser gets pregnant first. And if you'd like to finish the comic strip, because I really don't want to get in trouble with the school, scroll down here. Wait, where is it? Right there. Okay, so feel free to finish the comic strip on your own. But with flatworms, you can cut them up into tiny little pieces, and they will each become an individual organism on its own. Now, I'm also going to show you a YouTube clip of penis fencing. Believe it or not, Spike TV kind of did a special on it, but I can no longer find it because Spike TV is now owned by Paramount, and I'd have to pay for it. But I did find a National Geographic on YouTube that I will show you. All right, so penis fencing. Moving on. Okay, so there are 4 classes of flatworms, and your book does go into depth with them. Oh no, hold on. I don't like this here. Presenter view. There we go. So, yeah, 4 classes of flatworms. Make sure you check it out. There's the tapeworms, the tuberella, the monogeneria, and the Nematodes. Okay, moving on to pseudocoelomates, which means fake body cavity. We do have a cavity between the mesoderm and the endoderm, but it's not completely covered. Who's part of the pseudocoelomates? The ecdysozoans, specifically the nematodes, and the Plato Zones, which is the rotifers. So I'm kind of down to the end of getting really close to the end of my lecture here, just to talk about nematodes and rotifers. But the pseudocell, or the fake body cavity, if you will, kind of serves as a hydrostatic skeleton. And so it uses fluid under pressure to kind of gain rigidity and support, as well as some movement. It does lack a defined circulatory system. Now, the nematodes include roundworms, eelworms, and vinegar worms. The structure, again, bilateral, symmetrical, unsegmented. Okay, very important key characteristic there. Flexible, very thick cuticle, forms a hydrostatic skeleton, lacks respiratory organs, so they exchange oxygen through their cuticles through diffusion. And the mouth has sensory organs with some Pearson organs as well, called stylets. So you can find the nematodes on page 678 to 680 in your books. So just a general structure of the roundworms here, the nematodes. And so you can kind of see that the body cavity is not completely surrounded by a mesoderm, and you can see that these muscles here are being anchored to show a hydrostatic skeleton, and it kind of like works as, I don't want to say a pulley system, but you know, these muscles could like contract, and then it causes the organism to turn. Okay, so kind of a better diagram, if you will, showing the hydrostatic skeleton of roundworms. Okay. All right, the last phylum is the Rotifera. Okay, bilateral, symmetrical, unsegmented pseudocoelomates. These guys are very small, 3 cell layers, highly developed organs. It does have a complete gut. So it has a mouth and an anus. I would say one unique factor is how it gathers food with the mouth here. It's got this opening called the corona, filled with cilia, and they just wiggle, and food moves into the mouth and passes through a complete digestive system out the anus. So they are aquatic organisms. They propel through the water by beating their cilia, but then the corona is where food is being gathered. And so it's almost like a jaw. It kind of opens and closes to kind of bring food in. So Rotifera, what it looks like, cartoon versus real life. And I know I went through a lot here, so make sure you check out those page numbers in your books. But we just discussed the sponges, the Cnidarians, the Ctenidophoras, platyhelminthes with flatworms, Vertebrae, the Lophotrochozoas, the nematodes as well. Oops. Actually, no. This is in your book, though, so make sure you check that out. But the nematodes. And so then we will continue on and break it up into two parts where we'll discuss the mollusks and the annelids, the echinoderms, the arthropods, and the chordata.